Very good. Hello for this afternoon session. I'm coming from Estonia. My name is Elin, and I will introduce you Estonian Biobank, if you haven't heard about that, and also the research, actually what we're doing in Tartu, University of Tartu, where I'm working, and where we use this Estonian Biobank data, and I will talk to you what kind of research we do in order to move towards to precision medicine. So just a couple of this is disclosure, and then some introductory slides about our institute. We have a research institute, but we do a lot of research, we do also teaching, but we also host Estonian Biobank, and we use this Estonian Biobank data to do research. And we have several kind of research institutions, Estonian Genome Center, where I'm from, and we mostly use Estonian Biobank data, but also Estonian Biobank, also Biocenter and core facility, and so on. So I would say that we have been using Estonian Biobank. It's 25 years old, and we have been using this data already 25 years. We have been involved more than 300 scientific projects and published more than 800 papers about this research using this data set, and we every year doing a lot of research and publishing more, and we have many focusing using these data sets. And I will introduce a little bit later about these data sets availability, but for example, we focus in clinical genetic studies, working with common and rare diseases. We have functional genomics, also pharmacogenomics, mental health issues, and also microbiome, which is actually my research focus in this institute. So why we are working with that? We would like to really use the genetics data, and the idea is to kind of moving steps forward to precision medicine. And we know that we can use the genetics data to analyze the monogenic diseases, which are rare in population, but also the mostly common are common diseases, which is kind of diseases we know whose development is necessary many genetic loci, and also the lifestyle is important. And we are also focusing very much on pharmacogenomics, which is kind of what kind of genetics predisposition you have, how you tolerate the different drugs. And also in a biobank, in the 5 last years, we have been putting a lot of efforts to study not only our genes, but also the microorganisms. We live together, millions of microorganisms who has their genomes, and this is also affecting our life, and we are putting efforts to study this as well. So now I'm moving to introducing you Estonian Biobank. And it's a population-based, volunteer-based biobank. So we have currently more than 200,000 individuals collected from Estonia, and it's covering about 20% of Estonian population. So population in Estonia is small, 1.3 million people, and that's why it makes this biobank quite unique in terms of the other big biobanks, because it's covering quite huge amount of population in general. And this gives us possibility also to translate this genomic research to more to medical system. And these slides showing you the time scale since the idea bring up in establishing this biobank in Estonia. It's the 25th anniversary in a biobank in Estonia, and we have kind of 2 different waves. So the first data set was collecting around 10 years ago, and it takes 10 years to collect 50,000 individuals in the first wave, and this was a little bit different in the later collection. So it was involved all the GPs, around 500 GPs across Estonia. So the population that the whole country is covered, and we had a very deep questionnaire, also covering a lot of lifestyle and environmental factors. And this first wave, we also asked many questions about health-related issues and also the relatives, what kind of diseases they have and what kind of drugs they are taking. But we did a lot of research, and this was like initiated by the government to giving more money to us. And in 2018, we were able to start to collect additional samples. And within 2 years now, we were able to collect 150,000 samples. And this kind of recruitment was much easier way. We didn't use the GPs, but we instead of that, people who wanted to join with a biobank, they had to digitally sign approval to be in a biobank. And also we did like reducing the questionnaire in a biobank. We only ask more lifestyle and environmental data, and the health data we got from electronic health records linking. But there is also many other data sets which are available, and I would say that one of the most important point was to establish this kind of biobank was to really create a law. And it's Estonian Human Gene Research Act, which was the basis. And this Research Act gives a very broad consent. So all the people who enjoyed the biobank, they gave a quite broad informed consent, which means that we can always return back to each participant and design new study, collect a new data set, and also makes the transferability like to also ask people's opinion about the biobank. So this is kind of representing age and sex distribution in a biobank right now. And you can see that in terms of the age groups, we have covered most of the age groups starting year 18. And also when you see that the more females than males, and it's kind of general in a biobanks that the more females are volunteered to join with a biobank than the males. And there is also the second figure representing what kind of data we have available. So we have a lot of phenotypes data available, and it's most of the data currently, the health data coming from the electronic health records. But we also have, like I said, sorry, I'm just going back, maybe here. So a lot of questionnaire data as well, and also many different layers of omics data. So what we collected when people joined with a biobank, we always collect the blood. So we have a DNA available, and we have a plasma samples available for all 200,000 individuals. And currently we also have all the people, 200,000, we have genotyped. We have a subset of individuals, like 3,000 with a whole genome sequencing data, also proteomics data, also metabolomics data. It's now covered the whole population. It's a metabolomics, not the untargeted, covering the whole population, but using this NMR-based metabolomics, which is done for a whole cohort, but the subset of cohort, some thousands of samples, we have also untargeted metabolites information available. And proteomics, as I mentioned, and also, as I mentioned, we also have a microbiome cohort for a subset of individuals, 2,500, which collected 5 years ago, and we recently also collected a second time point data for microbiome. Since microbiome is affecting many factors, and we can't just use one probe to study the whole life, but we have to really collect the samples and see the dynamics and changes of the microbiome or organisms. So this slide's actually presenting more visually what kind of data we can get from electronic health records data. I didn't put here the more details that what and where, which kind of electronic health records, we get the data from different registers like cancer registers, myocardial infarction registers, population registers, death registers. We have connected health insurance information about what what kind of treatments people get, and also the diagnosis, and also we get the data from clinical lab measurements from 2 major hospitals in Estonia. One is Tallinn and one Tartu clinical hospital, and we also have access to full free text records. And here it's just representing what does it mean. It's just one person, and if we collect the samples here, we can retrospectively see what kind of diseases those persons have in their electronic health records databases, and what kind of treatments this person will get. And for example, if we get additional samples, like for example microbiome samples, we can also see what happens after sample collection. So it makes there really possibility to do several kind of different studies. And the next slide just presenting one of these, availability, how to study, for example, drugs. And here I just put in the slides about the microbiome recent studies which we did. We were really looking about the different drug effects to the microbiome, because we know that many drugs basically affecting the gut microbiome. We all know antibiotics, which is well known, but we also saw in our previous studies that many common drugs like beta blockers, antipsychotics, proton pump inhibitors, type 2 diabetes drugs, they have kind of, we could say, similar effect as antibiotics. And we carried out a more specific study to really use the electronic health records data and ask many questions about the direct effect of the drugs, the time when the probe, like for example, microbiome has been taking. We study and we see what kind of drugs people taking and how this affect the microbiome. But we also can see the effects, the carryover effects, so that the people not taking a drug but has been taking drugs previously, and see how this affecting the microbiome, and also the additive effects. And also we can see when we have a different time points, we can see drug initiation effects, just showing and how the electronic health record data can be used in several ways. So why we are actually doing these studies is that the bigger aim is, of course, to kind of looking the ways how to prevent diseases, and doing a more precise diagnosis and treatments. And we all want to live more healthier society. And moving now, I think the next slides is more, I will talk about genetics, our host genetics, not the microbiome anymore, but host genetics, where we have been done the most research in our institute so far. And we have 2 kind of directions where I now more focusing and moving faster is how to define the polygenic risk scores, to find individuals with a higher risk in terms of the genetics, and this makes, helps to make better decisions for health behavior and diagnosis, as I already mentioned. But also one kind of area where we are now moving more intensively is a pharmacogenomics, how the personalized drug recommendations for efficiency and safety. And we know why we need to do this, because we know, I think it's the same in Latvia, that the population is really kind of eldering. We know that there is a prognosis from Estonia registered to see that in 20 years perspective we have much more individuals with a higher age, which means that we really have to prevent diseases, not putting so much effort to treat people, but defining these high-risk individuals as early as possible can help to make better decisions in earlier than disease occur in a population. We started a year and a little bit more ago a very, very big project, which is a multi-center project involving many, many institutions, like 5 institutions from Estonia and 2 institutions outside of Estonia. And the major aim, it's kind of center of excellence in personal medicine, and it's funded by European Commission as well as state. And the vision of this is to kind of merge all the expertise and also activities to move towards to personal medicine in Estonia. So, and this kind of big project, it's a busy slide, but what we do there, we really use data to generate more science, research, and create new tools. And here we use this biobank data, electronic health records, and also the genetics data. And I'm happy to say also that during this project we get the funding to also going deeper. I said that in Estonian Biobank we have a genotype data for all individuals available, but these projects allow us to go more deeper and fully sequence 10,000 individuals using the long-read sequencing and the PacBio technology. And also we are part of the 1 million Genome European Union 1 million project and initiative, and this also brings another data layer in this project. But instead of also doing this kind of new genetics tools, and we need to test them. So in this project we also testing those clinically relevant let's say algorithms and doing also clinical studies in this project, but also we are incorporating also the system where we can define the cost effectiveness and also to bring socioeconomic and impact analysis and also educate healthcare systems. So many organizations and hospitals are a part of this to make this kind of flow so that they're bringing together different experts and also giving in the end recommendations and also providing this data to also the industry and establishing new spin-off companies as well using the health data. So this is really big project, but also we started the last year in our institute the 3 Center of Excellence projects, and the first one, Center of Excellence, Personal Medicine, led by Professor Redik Mägi. He's kind of supporting the previous big project and putting a lot of effort and input there to do this high-level research. And then we have 2 other Center of Excellences, and one of the focus in Estonian Biobank has been recent years also to study well-being science. We have been creating a different surveys to study mental health and very deep questionnaires and also the personality, which we can also incorporate with genetics data. So this personal medicine Center of Excellence, they have different angles and also like we know this 4P, it's a predictive, preventive and personal and participatory, and it's also like bringing together different experts in this very big project. So one important thing which has been happened in an Estonian Biobank, it's also that we kind of returning feedback based on the genetics data to participants. And this allowed our law, as I said, that we can return back to the participants and also to say what genetics predispositions they have. And we have then done that very many years, actually, already 10 years. And there are several different projects where we have been doing this kind of piloting studies, which gives important knowledge to establish a platform to give the feedback for a whole population, whole Estonian Biobank population. And those small pilot studies covering mostly, for example, breast cancer mutation screening, which was, I think, covered through my colleague who gave a presentation today, early morning session, and also cardiovascular diseases and many others. But one of the biggest kind of pilot study, before I'm going to this gene portal, which we opened this summer in Estonian Biobank, was a study which was done in 2017, And this pilot study was, involving almost 3,000 individuals. And here we gave a feedback in for 2 different diseases, type 2 diabetes and cardiovascular diseases, about genetic risk calculations. And this study was done so that the people who wanted to know about this genetic risk, they had to come to institute and had a face-to-face discussion with a specialist to explain what does it mean, this kind of genetic risk which they had, and how this affect and what you can do really with these risk calculations. And as I mentioned already, in the early this year, we were open kind of second pilot study for a bigger scale and invited 10,000 individuals to participate to My Genome portal, which is kind of for all the Genome Estonian Biobank individuals. And in this year's summer, we opened this My Genome portal to every Estonian Biobank participants. And within the first 2 weeks, I think more than 80,000 individuals were going to this portal and checking what is there. So a couple of slides, I'm just presenting what we have in this My Genome portal. And it's a portal where every participant can go with their ID card, so they can only one who can see this information. And it's very user-friendly and secure platform, and it has 3 different layers. One is the report, which we give, offering a personal genetics results. Also, very important part which we created in this portal is educational part to improve this, people knowledge about what is genetics, what are the genes, what we are studying, and what this data really means. And also, it's important to kind of provide a platform for research. So we, as I mentioned, that we can always return back to individuals. They can always be part of the next projects, and this portal giving the opportunity to see what kind of research projects are ongoing, and also what projects they can join if they want. So next slides, you can see that in this portal, as I mentioned, it's first giving some scientific data about the genetics cumulative risk of certain diseases. And we currently give genetic risk of coronary artery disease, type 2 diabetes. It's a polygenic risk score-based information. We also have a pharmacogenetics report about 28 different markers. We also give ancestry information, where you're coming, about the third generation information. And also we can give people what is the proportion of Neanderthal DNA is in your genome, which is very interesting, it turns out. Also the caffeine metabolism. And for example, here is example of type 2 diabetes. This portal is really active, so people can really play around. They can see what is their risk, and then they can play around, also putting their age, and also depends on the weight, and do they smoke. So because the complex disease is not the genetics alone is important, but the environment and your lifestyle. So you can really see how this risk is changing. And this is, I believe, the most final slide to show also that this portal was really covered in Nature, to saying that this one of the, the biobanks who are able to really give this genetic feedbacks to all participants, and some of the people's kind of expression, how they felt and what they feel to get this information. So we do a lot of studies. So putting this together, I think in Estonian Biobank, we tried to find those at-risk individuals in the really early stages to be able to predict diseases, to better diagnose and giving a better treatment, and not only using a human genome, but also, like I mentioned already, lifestyle factors and the microbiome. And just summarizing this, I think this Estonian biobank has really high quality, detailed and multi-layer phenotype data available. And this really helps to healthcare system and allows the framework for developing a personal medicine in Estonia. And also this, as I mentioned, the recontact of participants, offering this recall by genotype approach and collection of additional data set, and also study how people feel about to get this genetics information about themselves. So I think it's all, and I thank all my colleagues. These are big studies which we do, and many, many colleagues, and the biobank participants, of course, are part of the studies. So I'm happy to have questions if we have a time. Yeah, there is some.
Maria Trubitina, IQVIA Poland. Have you looked at the age distribution of people accessing the portal and whether it correlates to the donors' age distribution?
The answer is like all the ages, because half of them, right now I know that half of the people has been going to this portal and looking their feedback, so we were looking this age and it's all the age groups basically covered, yes.
That's impressive.
Yeah, people really waiting that, and some people were waiting 20 years, because I said 50,000 people were collected more than 10 years ago, even 20 years ago, and some people really, wanted this information a lot and asking constantly where they can get. And I would say that people, after this opening this portal, people who didn't join with a biobank, they started to call and ask, can we now join with a biobank? But they see the benefit of some, information, and that's I think the beauty of this portal. That the people willing to participate in research studies and develop on that. Yes. The biobanking team, yeah, that's in the research team, I think we have around 60 scientists. And some of those are more working in a biobank, keeping this biobank, and the research, I think around 50 researchers. Yeah. Okay, I'm available at least a couple of, I think, 1 hour, and you can talk and ask questions later. Thank you.